So what are these molecules? What is DNA? What is RNA? Why do they sound similar? Well, they are large biological molecules. And they are both polymers. And they're both polymers of nucleotides. And that is the general, nucleotide is the general name for the monomers. So if you recall, polymers are strings of repeating units called monomers, and monomers are generally small molecules that are similar to one another but not identical. And in both DNA and RNA, we have the monomers being nucleotides. In DNA, the exact type of nucleotide is a deoxyribonucleotide. And then in RNA, we have ribonucleotides. And so we can see where the R comes from in RNA and where the D in DNA comes from. And actually, we could also see where the N comes from in both of these. The N is for nucleotide, and then the A is actually for acid. So DNA is deoxyribonucleic acid, and then RNA is ribonucleic acid. That's what the RNA and the DNA stand for. And I'm going to write that up here. Deoxy. And I'm just going to break it down here so you can remember it. Deoxyribonucleic acid. Okay. So what do these molecules look like? Well, as we said, they are polymers that are made up of monomers. So we're going to look at both the polymer and the monomer. The polymer is here on the left side, and then the monomer is here on the right. So we're going to go through both of these. The monomers are nucleotides, as we said on the previous slide. And the monomers have three important groups. Again, as I said, the monomers are going to be pretty similar to one another, but not identical, meaning that there are different types, just the way we have different flavors of ice cream. All ice cream, chocolate ice cream and vanilla ice cream, they're pretty similar. They have different flavors, but the consistency is similar. They're cold. They have the same basic ingredients, et cetera, et cetera. So the similar ingredients in these monomers are the phosphate group, which is here, the sugar group, and then the nitrogenous base. So these are three different chemical groups that we're going to find in every single nucleotide, regardless of which type, which flavor. So some things that I want you to notice about each of these chemical groups. For group 1, the phosphate group, it has negative charges. And that's going to be important because it's going to give DNA overall a negative charge. And that's really important for how it interacts with other proteins, how it interacts with proteins, and also how we can manipulate it in lab. Then we have the sugar group here, and I want to point out that the carbons are actually not written in here. So this molecule, it has 5 carbons, and in order to count them, I suggest writing them in. So the 1 is here, then we have 2, 3, 4 is here, and then 5 is up here. This is going to be important when we start talking about strings of DNA or RNA and trying to distinguish the two ends from one another. And again, there are 5 carbons, and usually if I want you to look at one particular carbon, I'm going to give you the numbering for it. Finally, what's important about this nitrogenous base? Well, the nitrogenous base is what actually gives each nucleotide type its identity or its flavor. And so these are A, T, G, C, and then U. So these are the different types of nitrogenous base that can be present on a nucleotide. And this is what we normally focus on when we're talking about a sequence of DNA or a sequence of RNA. We usually just give the identity of the nitrogen space because we know that the sugar group and the phosphate group are going to be the same between every single nucleotide. So now let's take a look at these and see how they appear or how they're put together and contribute to the polymer. And so actually I'm going to highlight these with different colors at this point. So these different groups appear in the polymer, of course. So the phosphate groups you see here, the sugar groups we see here, and then nitrogenous bases we're seeing here on the other side. So basically, nitrogenous bases are on the opposite side of the molecule as the phosphate group when you have it in a polymer. Now let me highlight the individual monomers for you in this polymer. So this is one monomer. This is another monomer. This is a third monomer, and then this is our fourth monomer. So this polymer is a string of 4 nucleotides, and we see they're identical except for the nitrogenous bases in them. The other thing that I want to point out is that the 2 different ends of this polymer are labeled as the 5' end and the 3' end. This has to do with the carbons in that sugar group. And so, as we saw before, the 5 carbon is at the top, and then the 3 carbon is over here. So the 5' end of the polymer is the one that has the 5' carbon that's attached to the phosphate group, not attached to anything. So there's nothing attached up here. There's no additional monomers. There's no additional nucleotide. The 3' end is the end where we have no additional monomers attached to the 3' carbon, which is down here.